Hello and welcome to the Food is Medicine podcast. In this podcast, we'll be discussing how food has a drastic effect on our health, for good or for bad, and how food can prevent many chronic diseases such as obesity, diabetes, Alzheimer's, and fatty liver disease. Now, this podcast is part of the MedPorium program and still maintains the MedPorium mission of bringing free medical education to all. Now, if you don't know me, my name is Jahongir Karim and I'm the co-founder of MedPorium. Now, for the first episode of the Food is Medicine podcast, we really want to understand, just get an introduction to nutrition, because there are some terms that you have to know throughout the series to really understand why the food we eat is very crucial. Now, we start by discussing macronutrients. Now, what are macronutrients? Well, macro means large or in large amounts, and nutrients are just things that benefit us physiologically. So macronutrients are nutrients we need in higher amounts, and these come in three forms: carbohydrates, lipids, and proteins. Let's start talking about carbohydrates. Now, carbohydrates, the word carbohydrate comes from carbon hydrate, and carbohydrates are consisted of one carbon, 2 hydrogen, and 1 oxygen. It's a 1-to-1 ratio, and that's why some carbohydrates like glucose, it's C6H12O6. You can see the doubling effect for the hydrogen. Now, speaking of glucose, glucose is a monomer of carbohydrates, and monomer means just one unit, and specifically it's called a monosaccharide. A monosaccharide is basically one sugar unit, and that's what glucose is, as well as fructose and galactose and ribose, but we're not really going to talk about ribose. We mainly want to focus on fructose, glucose, and galactose as our monosaccharides. Now, there are also disaccharides. Di meaning two, saccharides meaning sugar, so these are just two monosaccharides bonded together. And these include sucrose, lactose, and maltose. Now, sucrose is basically fructose plus glucose, lactose is glucose plus galactose, and maltose is double glucose, just two glucose bind together. Now, to bind two monosaccharides together, there's a process known as dehydration synthesis that occurs. And this is when there's an OH part of one part of the monosaccharide, and there's an H group, or just a hydrogen on the other, and they bind together to form water and form a glycosidic bond. So it's basically a bond together, and the byproduct is water. And this is why it's called dehydration synthesis, basically creating water, or taking water out to create a bond. Now, there are also polymers, and poly means many, so there's just many units of these sort of monosaccharides, and these are called polysaccharides. Poly meaning many, saccharides meaning sugars, so just many sugars bonded together. Now, the main polysaccharides that we need to be familiar with are oligosaccharides, starch, glycogen, and fiber. Now, what are oligosaccharides? Oligosaccharides are made of 3 to 10 monosaccharides, and these include verbose, raffinose, and I hope I'm pronouncing this one right, it's stachyose, which are found in beans and legumes. Now, many oligosaccharides, like the human milk oligosaccharide, or HMO, serve as a prebiotic, and prebiotic just basically means food for the bacteria. So these HMOs, which are found in breast milk, can help cultivate the gut microbiota and further stimulate the production of the gut microbiota, which in a future podcast episode we'll talk about why the gut microbiota is crucial for our health. Now, starch is a pretty common polysaccharide, and one of the names it's referred to is homopolysaccharides, and these are found in potatoes, grains, and corn. Now, the thing about starch is that we can metabolize it fairly easily compared to fiber. We can metabolize it pretty fast, and this can result in a higher glycemic index or higher glycemic response. Now, glycogen is basically animal starch, which means this is something that we store glucose in, etc. Now, starch in plants, that's a storage unit for plants. Now, like I said, glycogen is just stored carbohydrates, and it's usually in the form of glucose. So glucose is basically stored, and which is called glycogen, and glycogen can be used when we're in a fasted state or when we're exercising, in which that glycogen will be broken down to glucose, and it'll feed our entire cells, either the muscles that we're working out with or the legs that we're running with. Now, one concept you have to understand is homeostasis. And homeostasis is a process of the body just maintaining balance. And this is the reason why we sweat when we're hot and we shiver when we're cold, because we're trying to maintain our body temperature. Now, last out of our polysaccharides is dietary fiber. Now, what makes dietary fiber special out of most of our other polysaccharides is that we cannot fully ingest dietary fiber. So one may ask, why do we even need dietary fiber? Well, dietary fiber feeds our gut microbiota. That's basically their food. And when we keep our gut microbiota happy, it keeps our body healthy. There's this sort of idea where you have a second brain. You know how people say, just go with your gut feeling. Well, that's actually true because your gut is actually your second brain. And we'll get into that in the next podcast episode because it's really interesting to learn about the gut microbiota. It can really influence our mood, how we sleep, how we eat. It influences a lot about our physiology. Now, there's soluble and insoluble fiber, and this is in context of being soluble in water. Now, the most common soluble fibers are pectins and gums found in oats, peas, beans, apples, and more. Now, the main insoluble fiber that is good to know is cellulose. And cellulose, we can't fully digest it. It's basically from the plant wall, the cell wall of the plant. And even some of our gut microbiota, some of our gut bacteria can't even ingest it. So what cellulose does is it basically pushes food around the digestive system. So we can't really ingest it. We can't digest it fully. Some of our gut microbiota may not be able to. It just pushes food around. Now let's move on to lipids. Now, lipids, also known as fats, comprise of one carbon, one hydrogen, one oxygen, and sometimes a phosphorus. Now, we have that 1-1-1 ratio. So, unlike carbohydrates, which is the 1-to-1 ratio, we just have a 1-1-1 ratio. So, there's going to be one carbon, hydrogen, oxygen, and sometimes even phosphorus. Sulfur is also included there as well. Now, the monomers for lipids are glycerol and fatty acids. Now, one thing to note is that the amount of carbons on the fatty acid correlates to how soluble it'll be. So if there's more carbons, it'll be less soluble. Now, fatty acids can be described as saturated and unsaturated. And this saturated part basically means that there's a lot of hydrogens around the whole fatty acid. And unsaturated means they don't have all of those hydrogens. That's basically what it is. And unsaturated fats can have a double bond while saturated fats don't. So they kind of contradict each other, kind of the opposite. And here's a picture showing both of the comparison. Now, unsaturated fats are broken down even further, and there's monounsaturated fatty acids. And monosaturated fatty acids are just fatty acids with a single double bond, or also known as a kink. And the polyunsaturated fatty acid refers to having more than one double bond, or more than one kink, maybe even multiple kinks between the hydrocarbon. And in general, long fatty acids that are more saturated will have a higher boiling point. And this makes sense because if we're saturated with a bunch of hydrogens around our whole fatty acid, it's going to be harder for heat to break those bonds down. And that's especially since there are hydrogen bonds. And this is similar in water. This is why water has a high boiling point. There's maybe like 211, 212. But this high boiling point is because there's so many hydrogen bonds. And although there's a lot of heat and hydrogen bonds are weak, there's just so many of them to break down. This is why water, like I said, has a higher boiling point. And this is similar to fatty acids, has more of those hydrogen bonds, so it's going to take longer time to break down those hydrogen bonds. Now, unsaturated fats can have a cis arrangement or a trans arrangement. Now, the trans arrangement is basically when they're opposite, when they're opposing each other. When the cis arrangement, they're basically on the same side, basically on like this, and then trans is kind of like this. The trans is going against the opposite sides, while cis is on the same sides. Now, the thing about the cis arrangement for the fatty acids is that it kind of bends, kind of like a V shape sometimes, when the trans phase doesn't have that bend, that sort of V shape. Now, the polymers of lipids include triglycerides, sterols, and phospholipids. And the sterols basically include cholesterol as well. Now, triglycerides are made of glycerol and 3 fatty acids bonded by ester bond. Now, phospholipids are made of glycerol, 2 fatty acids, and a phosphate group. And this is when the phosphorus comes in, like we discussed, in one of the elements. Now, phospholipids are amphiphilic. Hopefully I'm pronouncing that correctly. But that basically means that they can be both hydrophobic and hydrophilic. Hydrophobic means water-fearing, and hydrophilic means water-loving. Now, in the phospholipid structure, the hydrophobic portions are going to be more inwards in the cell membrane. They're going to be more inwards, while the hydrophilic parts will be more out. It kind of looks like this. These are kind of like what phospholipids look like if you could do it with your hands. You have the hydrophobic portion right over here, right in the middle, and the hydrophilic portions out over here. Now, sterols are lipid molecules that have sort of a ring structure. And the main sterol that I'm sure everyone knows is cholesterol. Whether you think cholesterol is good or bad, we'll talk about in the next podcast episode. But cholesterol is essential for human physiology. It's responsible for creating a plethora of hormones such as testosterone, estrogen, the androcortical hormones like cortisol, and etc. It's really important for human life. It's also, cholesterol helps maintain the fluidity of our cells. And this is very crucial for the cell membrane to bring things in and take things out so they can change shape, and this can benefit us physiologically. So it's very important for cells to stay fluid instead of rigid. Without cholesterol, our cells would stay rigid, which would be terrible. Our cells wouldn't be able to bring anything, and we'd essentially be in a bad spot in our health. Now, next thing we need to talk about are lipoproteins. And lipoproteins are basically transport vesicles for cholesterol. You can think of lipoproteins as a car and the people as a cholesterol. We need the car to take us all around wherever we need to go from point A to point B. Now, the thing about cholesterol and triglycerides is that they're usually hydrophobic. They cannot really mix with water, and in our bloodstream, they cannot be transported to where they need to be. And that's why we need lipoproteins. Lipoproteins are consisted of proteins, cholesterol, triglycerides, and the membrane around it usually consists of phospholipids. Now, there are 4 types of lipoproteins. The first one is a chylomicron, and this is the largest lipoprotein, but it's the least dense. The second one is very low density lipoprotein, or VLDL, and this transports endogenously produced triglycerides and cholesterol from the liver. Now, the third one is low density lipoprotein, or LDL, and this is a metabolic product from VLDL through successive hydrolysis of VLDL, the core triglycerides, which results in cholesterol-rich LDL. Basically, LDL is derived from VLDL. Now, the fourth type of lipoprotein, our last one, is the high-density lipoprotein. And as the name implies, this is the highest density lipoprotein. Although small, it contains the most proteins out of all the other lipoproteins. The lipoprotein that contains the most cholesterol is low-density lipoprotein. Now, as you can see from the chart, going from the chylomicron all the way to the HDL, the size decreases, but the density increases. Now, let's talk about protein. And protein is derived from the Greek word proteos, meaning primary. Now, proteins are made of a carbon, hydrogen, oxygen, nitrogen, and even a sulfur. Now, there are so many functions of proteins because proteins are very important for our physiology. For example, our internal tissues are made of collagen, our hair is made of keratin, the antibodies to fight infection are also made of protein, and the creation of enzymes are basically are made from proteins, and it is necessary for building muscle. So protein is very essential for our physiology. It's just so important. Now, the monomers for proteins are amino acids. Now let's look at the structure, sort of the anatomy of the amino acid. First, we have a central carbon, and then we have an amino group, which is just NH2. That's where the nitrogen comes in. And then we have a carboxyl group. Now we also have an R group, and the R group basically gives characteristics to our proteins. The protein could be hydrophobic, hydrophilic, it could have less of an electron, more of an electron, etc. It could be negatively charged, positively charged. So the RH group basically gives characteristics for the protein. Now the polymer for proteins are polypeptides, which contain multiple amino acids in a long chain. Now let's dive into how proteins are really made, going from amino acid to a fully developed protein. Now we start with the primary structure, and the primary structure is basically a polypeptide, just multiple of those amino acids just binded together. Now the next one is secondary structure, and this is when there's hydrogen bonds created between the polypeptides, and this results in the spiraling of the polypeptide or folding of it. Now, next one is a tertiary structure, and this is when the 3D sort of shape forms, and this is when the RH groups interact. If they're hydrophobic, they'll move more inwards, hydrophilic, more outwards, etc. So this is when the RH groups interact, and this is where the protein's characteristics really come alive. Now, the next one is a quaternary structure, and this is when there's two or more polypeptide structures after the tertiary structure. So they basically come in together and just form a nice protein. One of the examples is hemoglobin. Now, hemoglobin is a protein in our red blood cells, and from the tertiary structure, there's a beta protein, there's alpha protein, maybe there's 4, I really can't remember, but they basically come together and form the protein, the complete protein. Now we'll talk about proteins in a future episode because it's really crucial to understand. There are also essential amino acids we'll discuss and how proteins help us build muscle. Now let's move on to micronutrients. And micronutrients are nutrients that we need in a small amount. We don't need it in large amounts. And these include vitamins and minerals. Now, vitamins are defined as something that the body cannot produce itself. Vitamins are broken down into water and fat-soluble. Now, fat-soluble vitamin digestion is dependent on bile production, and bile is basically a fluid secreted by the gallbladder, which contains things like cholesterol, other fats, salts, etc. Fat-soluble vitamins also depend on the delivery of bile into the duodenum, which we'll talk about in the digestion and absorption of specific foods. Now, fat-soluble vitamins include vitamin D, vitamin A, K, and E. There's an acronym that I saw on YouTube, and it was a really good one. It's basically to remember their fat-soluble vitamins. It's Drake, but without the R, so DAKE. Vitamin D, A, K, and E. Now, water-soluble vitamins, as the name implies, are soluble in water, and they are involved in energy metabolism and coenzymatic activities associated with metabolic pathways of macronutrients. But you just have to remember that they're just water-soluble. This also basically means that they help in metabolism. Now, we will dive into more detail about vitamins in a later episode, but the main water-soluble vitamins are B6, B12, vitamin C, thiamine, riboflavin, folic acid, biotin, and pantothenic acid. Just remember that water-soluble vitamins are just soluble in water, and we'll talk about specific ones, but I think that should cover primary ones. Now, minerals constitute less than 5 to 6% of total human mass and can be divided by major minerals and minor minerals. And I'm sure you've guessed from the name, major minerals are something that we need in large amounts and minor, not that much. Now, it's really defined as major minerals are the average daily dietary need is over 100 milligrams and represents about over 0.01% of the total human mass. And these include calcium, phosphorus, sodium, potassium, chloride, and magnesium. Now, sulfur is also included, but its dietary need is still questionable. Although the WHO states that the recommended amount is 3 milligrams per kilogram per 24 hours in a healthy adult. Now, minor minerals represent less than 0.01% of total human mass. Now, these minor minerals include iron, zinc, iodide, copper, selenium, fluoride, chromium, and manganese. I think those are the primary ones. Alright, so now let's talk about digestion and absorption. Now, if you didn't know, digestion begins in the mouth. And this is when food enters the mouth and chewing, also known as mastication, occurs. Now, there are salivary glands around the mouth that secrete saliva, and this saliva contains enzymes such as amylase that help break down the food for swallowing. There's also lipase in the saliva that also help break down fats, specifically fats. Now, food is referred to as bolus as it travels through the esophagus through a mechanism known as peristalsis. And peristalsis is basically a wave-like movement. It's like, pretend if this is your esophagus, it's moving around kind of like this, kind of like a wave-like, and it's pushing the food down. So once food, or the bolus that we had from the esophagus all the way down to the stomach, enters through the stomach through the esophageal sphincter, and it is submerged into gastric juice in the stomach, which is just a mixture of hydrogen chloride solution, some lipase, and pepsin, all of which are just chemically there to break down the food. Now, once that breakdown occurs, the food is now referred to as chyme. Now, chyme will exit the stomach and enter the first part of the small intestine, which is known as a duodenum. And here, pancreatic juices are secreted, which contain enzymes such as trypsin, lipase, and amylase. And the liver will produce bile and store that bile into the gallbladder. And the gallbladder will secrete the bile through the common bile duct into the duodenum. Now, pancreatic juices will be secreted through the pancreatic duct, which joins into the common bile duct. Now, one thing to note is that pancreatic juices mainly break down carbs, proteins, and fats, are our main macronutrients, and bile primarily breaks down fat. Now, in the small intestines, absorption occurs by enterocytes, and enterocytes are cells that go around the intestinal lining, and on top of them, they have small finger-like projections known as microvilli, and the enterocytes will cover an entire villi. So it's basically like this. You have your enterocyte right here, and then you have the little microvilli, little projections right there, and those little enterocytes with their microvilli on top will surround the whole entire bigger villi, which is the bigger finger-like projection. Now, the bigger villi, the one that is the bigger finger-like projection, contains blood capillaries for the absorption of glucose and amino acids, and lacteals for the absorption of fats. Now, this goes to show how important the lymphatic system is in the absorption of fat, because the lacteals literally go into the lymphatic system. Now, you may be wondering to yourself, why do we have microvilli and villi? Why do we have these finger-like projections? Well, the main thing that they do is increase surface area. And this is crucial because they can bring in more nutrients, they can bring in more glucose, amino acids, etc. through a smaller amount of space. So they just bring more surface area. Now, food from the small intestine will travel to the large intestine and turn to feces through water and electrolyte absorption, which is some of the functions of the large intestine in digestion. Now, in the large intestine, there are over 400 different species of bacteria which make up the gut microbiota, and there will be an episode in the future dedicated towards the gut microbiota. Now, specific locations is just mostly around the entire gastrointestinal tract, but they're primarily in the large intestines, sometimes even small intestines. And after feces is produced, I'm sure you know what happens next. So, that pretty much concludes digestion and absorption. And in future podcast episodes, we'll discuss specific digestion and absorption of carbohydrates, proteins, and fats. Now let's talk about the big chunk of nutrition, which is talking about calories, metabolism, and energy. Now, we first need to define a calorie. Now, we all know what a calorie is, but what is the true definition? Because not many people can really answer that question. And it is a unit of energy equivalent to heat energy needed to raise a temperature of 1 gram of water per 1 degree Celsius. Now, that's the more formal definition, you could say, but it's basically the amount of energy a food or drink contains. Now, for carbohydrates, we get 4 calories per gram, as well as proteins, we get 4 calories per gram. But for fats, we get 9 calories per gram. So fats usually have more calories. Now, carbohydrates, proteins, and fats also have different functions in terms of energy. Carbohydrates give us short-term energy. Proteins don't give us a significant amount of energy, not a lot of energy unless it goes to a process known as gluconeogenesis, which we'll talk about later, and fats give us long-term energy. So this is why some athletes before their sporting event will eat maybe a bowl of pasta or some bread before a sporting event just to get some carbohydrates in them, just to get a short-term energy. This is why sometimes in bodybuilding, there's a pre-workout meal, which comprises a good amount of carbohydrates just to give enough energy to last for the workout. Now let's define metabolism. Now we kind of understand metabolism, but it really encompasses every chemical reaction in the body. But for this podcast, we'll refer to metabolism as a conversion of food to energy. Now, metabolic rate is the amount of energy expended or burnt in a given period of time, and the basal metabolic rate is the amount of energy used at rest. In the context of metabolism, the body will metabolize proteins to amino acids, sugar to glucose, and triglycerides to fatty acids. And those are all examples of catabolic reactions. See, we're breaking down proteins to amino acids, we're breaking down the sugars into glucose, fructose, etc., and we're breaking down the triglycerides into fatty acids. Now, if you know anything about health, you know the phrase calories in and calories out. And this is a basic thermodynamic idea in which energy cannot be created or destroyed, but it can be transferred and conserved. Now, I'm sure you've also heard of the body mass index, or BMI. And although it is sometimes flawed, it's one of the best measurements we have to determine good health based on height and weight. And over 25 BMI is considered overweight, and over 30 for BMI is considered obese. Now, one of the basic understandings of being overweight and obese is eating too many calories that you expend. So basically eating more than you burn. Now, this is true, but some calories are not created equal. Think about it. If you have 1,000 calories of broccoli and 1,000 calories of Coca-Cola, both of them are getting metabolized very different. They have the same amount of calories, but they're still metabolized very differently. For Coca-Cola, for example, it's much easier to drink 1,000 calories of Coca-Cola than eating 1,000 calories of broccoli. And there's nothing to really delay the metabolism, so the body doesn't need to expend much energy to metabolize all of that Coca-Cola. It just goes into the body through the liver and just metabolizes the fructose, and that can be really bad in the future, which we'll talk about later. But the 1,000 calories of broccoli contains fiber. In the presence of fiber, since we know it's indigestible, it makes it harder to basically digest that broccoli and take all the nutrients that we need. So we expend more energy to break down that broccoli than just drinking 1,000 calories of Coca-Cola. And that fiber, we know it's crucial. So I mean, it's pretty much impossible to eat 1,000 calories of broccoli, but you get my point. Broccoli also has a lower glycemic response compared to Coca-Cola. Coca-Cola will spike the blood glucose levels, while broccoli will keep it steady, maybe a little bit, not too much of a spike, but it will keep it relatively steady. So that's the thing about calories in versus calories out. It is true, but sometimes we don't really account how different foods can be metabolized by the body, how some energy can be expended, because 1,000 calories of Coca-Cola is very different from 1,000 calories of broccoli. Now, I'm not opposing the idea of calories in, calories out, because it is crucial and it is crucial to understand, but we also need to understand that some calories are not created equally. They're metabolized by the body differently, and we need to constitute that as well. Alright, and that wraps up our first episode of the Food is Medicine podcast. And this was basically an introduction to nutrition. And as always, a Pomodoro day will make you a doctor someday. I'll see you later. And that is especially since... I keep doing this. Oh, Jesus Christ. Damn, this is not easy. I get paid enough to do this.